Okay, in this lesson we'll continue our journey towards understanding reproduction and heredity. In this lesson we'll examine the chromosome theory of heredity. So to recap where we've been, we've just learned about Hammerling's research. And he was using acetabularia to investigate the role of the nucleus in regeneration. Regeneration is similar to reproduction in the sense that the cell needs to have information and building materials to reconstruct the missing part, just as parents have to have information and building materials to make an offspring. Well, in the case of acetabularia, here we see the foot of the organism, and here was the nucleus. And we learned that the nucleus produces molecules that leave the nucleus and enter the cytoplasm. And these molecules are information molecules, not building materials. The experiments with radioactivity indicated that when you make these nuclear molecules radioactive, they remain in the cytoplasm. The radioactivity remains in the cytoplasm when the cap is regenerated. So the cap, it was made from non-radioactive materials, which suggests that these nuclear molecules are information molecules that have the information to build the proteins that will actually go into constructing the cap. Today we know these molecules as messenger RNA molecules, and they are a temporary copy of information stored more permanently in DNA. So we can think of messenger RNA as a recipe for building a protein, and it is a copy of a more permanently stored recipe that is contained in the molecule of DNA itself. In this diagram we see a paramecium, and here's the nucleus. And this diagram will serve as a little review about the relationships between some of these molecules, proteins, messenger RNA, and DNA. First, let's take a look over here. We have a food vacuole, and you'll recall that we studied a couple different proteins that do different jobs in processing food for paramecia. So here we have bacteria coming in through the oral groove into a food vacuole, and in the food vacuole we'll have digestive proteins going to start breaking up the bacteria into smaller nutrient molecules. Those molecules would not be able to cross the membrane were it not for transport proteins. So the transport proteins will shuttle different types of nutrients out into the cytoplasm. Of course, one of those nutrients would be glucose. It will be attacked by a team of proteins to release energy, etc. So here we have, though, two proteins that are involved in digestion and food processing. Well, each of these proteins then has a corresponding recipe, a corresponding messenger RNA molecule that has the recipe for how to build that protein. The messenger RNA molecules are leaving the nucleus, coming out into the cytoplasm, where they will hook up to protein-making factories called ribosomes. You'll learn more about that in 9th grade. It is here where the information in the messenger RNA molecule will be used to precisely build the protein. And when proteins have different functions, that's because they have different shapes, and that's because they have different sequences of amino acids, the building blocks of proteins, and so correspondingly they will have different messenger RNA molecules. So there are different recipes for different proteins. Now each messenger RNA molecule then originates in the nucleus, and we'll be learning about this molecule of DNA, which resides in the nucleus. It is the sort of master set of recipes for building all proteins in the cell. Messenger RNA molecules will be temporary copies of the information stored in DNA. So at this point then, we've learned a little bit about the nucleus. It controls cell activities by producing important information molecules. These information molecules are the recipes for building proteins, which of course are the workers of the cell. And now we're going to turn our attention to what's in the nucleus, the origin of these messenger RNA molecules, the DNA molecules themselves. A long stretch of DNA that is wrapped around proteins will be called a chromosome. So in a sense, we're seeing a little part of a chromosome here with two regions of the chromosome having the recipes for two different proteins. We're going to, in the future, give the name a gene for a sequence of DNA that carries the recipe for a protein.